Hello. Hi, Mainak, and Tharun, and Dick, and Akshay. Hi. Hi. Wait a couple more minutes for people to come into the meeting. Other than that, welcome to the meeting. Did anyone have any questions before we started? Or does anyone want to present something? If they've been doing something with Google Summer of Code, would you like to present? Okay. So why don't we ease into the meeting here? First, I'd like to point out that hello, Aayush. First, I'd like to point out that I submitted an abstract to the INCF assembly, and we got accepted for a talk on the DevoWorm platform. So let me share my screen here. Okay. Hello, Abhishek. So this is the INCF assembly. This is planned for April 19th through the 28th. This is going to be organized as a virtual meeting, so it's online. And of course, the abstract submission is closed, and we were accepted into one of the sessions. So this is a session on the DevoWorm platform. I think I showed the abstract to people last week. The registration is fairly reasonable if you want to attend. At least if you're a student. I don't know what reasonable is to people, but you can. And so if you're not familiar, if you're new to INCF, they have a website here. It's the International Neuroinformatics Coordinating Facility. And so they're enabling a lot of open science, and they actually sponsor the GSOC program that we participate in. And so I'm presenting to them. I think they'll like the fact that I tied that into the talk. And this didn't happen last year, but it happens every year usually. It was in Warsaw, Poland in 2019, and I had a student who attended that Congress, and he actually did a demo of his software at that event, and he said it fostered a lot of interest in what he was doing, so that's good. So I don't know if they have a page for the assembly yet. Okay, this is it. So this is the assembly. It's a virtual conference, 19th through the 29th. They have, I don't know if they have a program ready to go. Okay, here we go. So, they have a bunch of sessions. They have one on what they call FAIR, which is a framework for sharing data for open science and open data. So it's like FAIR stands for findable, accessible, interoperable, and reproducible. And so the idea is to have this set of criteria that follow these guidelines for sharing data, or actually reusable, but reusable is sort of similar to reproducible. So this is a session on this, if you're interested in those kind of topics. What else do they have? They have a lot of different showcases for things. They have a session on electrophysiology, session on computational neuroscience. There's some OpenWorm people involved in this. Sharon Crook is involved. She's sort of OpenWorm adjacent. Some other people. There's open innovation and intellectual property, data science and neuroinformatics. So yeah, it's going to be a pretty decent event. I don't know why it ends at the 23rd. I think they have events afterwards that go on for a couple days after that. But anyways, that should be an interesting event. I see Susan and Ujjwal and ABD and I guess ABD was here. Shruti is here. Okay. And Akshay was here. Okay. So hello everyone. I was just talking about the INCF conference or the congress that they have.
Yeah.
So let's see. What do we have in the chat? Shruti, thanks for the Mars calculation. Hi, Susan. So that's what was the Mars calculation?
Actually, Dick had some work, and for that he needed some help, so he mailed it to me and Krishna, so we wrote some code for that.
Oh, okay. That's nice. Yeah. Yeah, that sounds pretty good. Dick says, clay is 100 meters deep on Mars, which I'm taking as representative of Haiti and Earth, which of course is like just Earth of the distant geologic past. So they're working on a paper for the origin of life, so that's what that's about. And we have a sort of a separate group of people for that, so if you want to join in on that, let us know. Let's see. Did anyone? I asked this earlier, but only a fraction of you were here. Did anyone want to present today on their GSOC proposals or anything that they were working on?
Yeah, I have 2 things to present.
Okay.
Yes. I presented it.
Okay, why don't you go ahead and do it?
Okay, so I should. Just a second. Yeah. So first I'll present the cellular automata thing which I'm working on. Last time when we met, I told you that I was working on cellular automata. It is just in simple words saying that we can create the simplest possible complex system for various problems in the world. Last time, what I discussed was the basics of cellular automata. Today, we'll go one step further where I have implemented the elementary cellular automata, where what I did was using the basic Python code and using different rules, like here, the example which I'm showing is for rule number 30, and a very basic code. Using that, we can generate what would the output of cellular automata will be. Using the same code, I've generated all these rules like the rule 90, rule 87, rule 202, 126 and all. This is very intuitive, and I guess if anyone is trying to understand something about cellular automata, just a suggestion to code it yourself, because then in that way you come to understand a lot of things about the rules and how the cellular automata and all these rules work. So this is the first thing that I did, and the second part was that I till now this was the general elementary cellular automata, and this is a 1D cellular automata in which we have a single row, and based on that row the next generation is made. But Game of Life, which is based on something which John Conway did, is a 2D cellular automata. And just to take my understanding to the next level, I tried to work on that too as well. By the way, if anyone wants the code for the previous this code, you can use it, and I guess it would be really helpful for you to understand what different cellular automatas with different rules look like. Yeah, so the Game of Life was basically created keeping biology in mind, how from a very simple organism like bacteria and the whole thing, the whole life on Earth started. It is what it is trying to replicate, and it does it very beautifully. It passes the Turing test, and stating that yes, a very simple algorithm like this can imitate the complex environment around us. So this apparent game is a zero-player game, meaning that its evolution is determined by the initial state, and we don't require technically any player here. Just the initial state from where where our model starts. It automatically learns based on some rules, which I'll tell you later, very shortly. And using those rules, we can literally develop and try. We can see how the whole environment works. So the rules that I was talking about are very generic. For the 1D automata, cellular automata, there were like 256 rules because there were 8 combinations that were possible. But here, what is it that we have for a single cell? Since it's 2D, so the number of neighbors for each cell would be 9. And hence there will be 512 possibilities of rules, and having so many rules and calculating based on them would be like really tedious. So what we have generalized the rule in the game of life, and we have just put it down to 3 things, that is the death, birth and status. Death of the cell means that the state, oh I'm sorry, I wrote it wrong, it is from 1 to 0, that is when it was alive, and now it is 0, that is it's dead. That happens because of 2 conditions, overpopulation and loneliness. Like in general life, when there's too much of anything or too less of anything, we have not people die, but actually the cell here dies. Where the overpopulation means that if the cell has more than 4 active members, that is, the neighbors here are, the more than 4 of them are number 1, then it would die. Or if they are less than 1 or 0 number of active neighbors would also lead to the death of the cell. Birth would be when there would be exactly 3 neighbors, and status would be nothing would change, it will stay static, and there won't be any death or birth here, like you can see there. So when you implement these rules, some really fascinating patterns occur. Some are static and some are not. For example, the top ones which you see here are like the block, the V-hat, the low, the boats. These are the static ones and they do not change. The next one is this one which keeps on oscillating to back and forth. So using all these combinations of from all this starts from a very single cell, and then it replicates itself with time. And I guess a picture is missing here. Nevertheless, so yeah, what happens is when you put all of this together in the form of a bigger picture on a grid, on a 2D grid, what you can see is a very beautiful representation of the real life world where things are oscillating, interacting with each other, and developing into newer things. Because of all these, the game of life is one of the very basic applications of cellular automata. It's something which is like the elementary things everyone does when they try to learn about cellular automata, but there are some very nice applications of it. The first one which you can see on the screen is a traffic flow control system in which the speeds of the car which is near or far, using all these parameters, you can literally see the traffic and you can, imitate it in the form of all the motion that you're looking at is in the form because of the cellular automata that is used. Another application was map generation using the cellular automata, in which we start with some random noise, and as we increase the number of steps, that's with each generation, you can see a pattern that's forming. The more the number of iterations, the more smoother the map would get. It can be very helpful in real life situations. This is another interesting thing which actually I found in the paper which was published by this organization itself. So it is about image restoration, where initially what you do is you have an original image and you do edge detection, and using those edges you can literally restore the image using cellular automata, which is really fascinating out there. Also, image segmentation, the main point of all this was because we are working on a project where we require image segmentation, and image segmentation using cellular automata is something that has interested the world recently, but not a lot of work has been done here. A very recent paper from Google AI, which was published in August 2020, is regarding this image segmentation by cellular automata, where they take the image and convert it, and using the image instead of just a neural network, what they take is a cellular automata to get for semantic image segmentation, and apparently it shows very good results. They have compared the neural network with the cellular automata, and they say that there is a very critical difference between the two. Like in standard neural network, what we have is a common picture, is a common intuitive picture that goes into deeper layers, like in deep learning. But in case of cellular automata, the deeper layers are actually the same as the earlier layers, and they just evolve with the state of it. That is, we don't have a new picture each time, but it evolves itself with time. Another way to think about it is to see the cellular model as a recurrent convolution network where the output of each sequence of loop is fed into the model. So such intuition don't capture the local organization, I get it, but this is something which is very fascinating, and I'll be looking forward to propose it for the GSOC as well, because this is a different approach which can add an edge to how we compute the things and maybe provide better results than probably deep learning, or if not better, at least they can be comparative to each other. Yeah, so this was what I had for the cellular automata thing. While doing my research here, I just found up something very interesting, a nice software. It is called the Cell Profiler software, which is a cell image analysis software. I talked about this to Bradley as well as Ujjwal. This can be like really helpful for our various bio images, because this is specifically for them. So this, first of all, some introduction. Cell Profiler is basically a free open source software. It can identify and measure biological entities. It can process images at any scale, at small scale, at large scales, and we handle it really well. And we can export the data for further analysis as well. It was basically developed by Anne Carpenter. She's from Broad Institute, and it is something that is being used very nowadays. It's in trend right now for bioimages. So some of the features where this software was something that caught my eye was because we can load from a series of image processing modules, and we already have some predefined, because since it's for biological images, it already has some pipelines for image segmentation, for labeling, in fact, not just for that, for nuclei, and I'm not very good at all this biology stuff, but yeah, for literally different aspects of phenotypes, I guess they are called. For different aspects, they have different modules, smaller modules, which we can work on. Also, we can adjust the settings according to our need. We can label it according to if you want the brightness to be more, to be less, separating. In fact, image segmentation was also something that can be done from here, and of course, it is built on flexible pipelines. Another feature of Cellular Profiler was the Cellular Profiler Analyst. It's not just about pre-processing the data, but we can also analyze the data using this software. We can explore the data. A very important feature of this was the data is interactively linked to images. So whenever we do some work on data, it directly is connected to the images, and manipulating stuff would be directly, whatever we manipulate would be directly reflected. It has various visualization tools which can help a lot, and like I said, each data point is linked to the image that is produced, and it has a classification tool as well, in which what we can do is, initially for some images, we'll have some particular n number of classes, and we can classify different images based on that this image belongs to class A, this to B, and after some time, it will itself learn like what are the different types of images that can work on that belong to different classes, and it can classify them on its own. So I guess this tool is something that can work really well for us, especially for bioimages, because it's particularly made for that. And I guess I will be working more on it and exploring more. I just discovered it yesterday, and I did not have a lot of time to go through it all, but I hope this will be something that will help us a lot in our projects. So yeah.
Well, thank you.
Okay. Can I make a comment?
Yeah.
Sure.
Here, I want you to look at something. This is a snail shell. Perhaps from that's it. Can you see it?
Yeah, you can.
Yeah, okay, you see the patterns are similar to some of the patterns.
Yeah, I have an image in the previous presentation. I showed this, and it's.
Oh, you did? Okay, good.
Yeah, it's beautiful.
okay, there's an unsolved problem here. And that is, how do we test whether or not the snails actually use these mechanisms?
Right.
Okay?
Yeah.
In other words, if you had a picture of the snail, can you not simply make a similar pattern, but can you extract the rules it uses? Okay. Okay. It's an inverse cellular automata problem.
Oh.
Okay. No one has tackled it yet.
Oh.
Yeah.
Okay. Okay, and there are many different patterns to analyze.
Hmm.
Okay.
I'm still interested in taking images of a shell in my microscope, but I'm at crunch time at school, so it's going to take me a few weeks. Okay? Can you hear me?
Yeah. Yeah.
Okay, good.
Okay, one other comment on the. Let me ask you a question about that cell analysis program. There are cells called archaea, which look like flat polygons. Okay?
Okay. Okay.
Given images of them, would that program be able to give you a distribution of the polygon shapes and sizes?
You're talking about the cell profiler or like
Right.
the soft?
Right.
I actually like I said, I will be exploring about it. I have not used the software to that great extent, but I think it should work like that. I can't tell you for sure it will.
Okay, I'll send you a picture after we're done, and you see if it works.
Okay, done.
Okay.
Yeah.
Yeah. Yeah, so I think this is great. Using a software like Cell Shaper is, of course, another alternative, and it has the advantage of not having to train the model before we use it. So with a lot of the machine learning models,
The data is a little easier in this the software. So because in I think before some 2 or 3 meetings before, Ujjwal was also telling that here we need to work on data manually because the data is not available that much. So I guess instead of just pointing, working it like literally pointing out each and every point, if we have a software like this, which is particularly for bioimages, I think it would be a great help for us. And data augmentation techniques too are there in this software. I can't comment more on that because I haven't used them right now. Maybe in the next meeting I'll let you all know what are the different things we can do it, and hopefully it will help us.
yeah, it would be great.
Yeah.
Then the cellular automata, where did you get that NumPy code?
I wrote it myself.
Okay. Do you have it on your GitHub?
Yeah, I will be uploading it and I'll share the link. Or we can upload it in DevoWorm as well because it would be like helpful for people whoever wants to learn about the cellular automata thing.
Okay. Well, that sounds good. Thank you for the presentation. Yeah, interesting presentation, Mainak said.
Thank you, Mike.
Did we have anyone else wanted to present today, or?
Also, one more thing. I'm like very actively looking for more stuff on cellular automata. So if anyone is having any resources or any recommendation, I would love to hear from you.
Oh. Are you aware of the Mexican group that pulls together much literature on cellular automata?
Not really.
No. Okay, I'll see if I can find it.
Yeah, that would be great.
Yeah. Great. All right. Okay, so yeah, I guess let's see. I wanted to give anyone else a chance to say, if they want to share something or if they have anything they want to. Okay, Ujjwal says, I'm a little skeptical about the results the software will produce, especially in the case of basilaria, where the cells are connected together. Look forward to results from this tool.
Yeah, I particularly was concerned about this too, but there in a tutorial which I saw, they had a thing where initially the cells they were connected to, it was not a bacillary cell, of course, I don't remember the exact, but they were nucleus of different cells that were connected really together. And as we improve the settings, we had to, it is something we have to do manually, like we have to see it, and so it was quite doing quite well in separating the different cells there. So I hope it will work good in this too. It's something that I hope it has.
Yeah. I mean, the thing to keep in mind with these approaches is that there are always pros and cons. So there are always things that some approaches do well and some approaches do poorly. So, to be able to go through and to evaluate that, the pro here would be maybe you don't have to train it, but the con might be that it doesn't do well on certain images. And then there might be a pro where it might actually do well on some types of images. And so it's, it's always that sort of balance between the two types of things.
Yeah.
Yeah.
I'll keep you both updated, Bradley and Ujjwal, and let's hope for the best.
Yeah. Dick put a number of links in the chat on Cellular Automata. So this is a bunch of different groups: Mexico, UK, Japan, China. So just labs around the world where they do this kind of work.
Yeah, I'll go through all of them. Thank you, Dick.
Sure.
Yeah. Okay. Good. So I'm gonna move on to, let's see. I'm gonna share my screen again. And I wanted to put a link for those of you who came late. I was talking about the INCF assembly, so I wanted to put a link in the chat on that. And again, this is something that I'm going to be presenting it on behalf of the group's activities, on behalf of DevoWorm and a couple other things. And we'll go through the slides next week, so I haven't quite gotten them together yet. And then this will be on the week of the 19th, I believe. So this is, look into it and see if you want to attend. Okay, so let me close that out. Next thing I wanted to do was, let's see. I did want to go over the onboarding guide. So the onboarding guide is this thing that, myself, Mayukh, and Krishna have contributed to build like this comprehensive guide to people coming into the organization. So if you're coming in as a GSOC aspirant, or if you're coming in as someone who just is interested in the group, this onboarding guide might help you find things. And we'll be adding to this continually. I wanted to make this very general so that it contains a lot of different things that we have. So you can see there's an index up at the top. And it's, maybe becoming a little too long, but I'm trying to... That's why I built this index, so that we have, if people want to go to different places in the, different topics, they can go there. So, one of the things that I've found in past years, and I haven't had a really good way to deal with, is people want to become involved in the group and they don't really have a background in biology, or they want to find some data sets to work on, or they want, in this case, they want to know about the projects. So this onboarding guide, I'm trying to build a lot of that into it so that we have it just at one link. I can send someone a link and they can read through it. So at the top we have an introduction to the OpenWorm Foundation, the DevoWorm group, some introductory slides on GSOC, open source contributions. Then some set of project descriptions. And this is where we have the GSOC projects. And this is, if you're interested in a project, you can go here, find some more information, some data. We're continually updating this. And so, next year we would put a different set of projects in here. And that's the way that would work. But still, even if you're not interested necessarily in GSOC, we have some good general FAQ for GSOC, but it's kind of maybe useful for other things as well. So we have I wanted to point this out to some people who asked me about the structure of their proposal. This is a section where, the question in the FAQ is, how do I structure my proposal? And I've given a couple points here. I'm a beginner developmental biology. Where should I start? We have a model organism section down here, just below. And that's where we have at least 3 model organisms. We have C. elegans, diatoms, and axolotls. And we should probably add more, but I didn't, for the immediate needs, we don't really need to add any more. But I'd like to add more. If we have resources we can put into here, it would be great. The resources we have now are basically like, different basic introductions to the biology of these systems. So for the Basilaria, I have a couple of links on diatoms. Thomas Harbeck's resource on diatoms, and a Scientific American article. So it just tells people about what diatoms are. C. elegans has some we have probably more references than this, but I've linked to the Worm Atlas, Stephen Larson's talk on openworm, some sample images of C. elegans, microscopy images, and then some basic biology and microscopy. And then for axolotl, a couple links as to, what it is and how it's important. So if you're doing a proposal, I would suggest, going through some of these links. And I know a couple of you have reviewed your proposals and you didn't really put a lot of emphasis on the problem that you're trying to solve. So maybe a fourth part of structuring the proposal is do a very brief introduction to the thing you're trying to study. So if you're applying to Project 1, for example, you would talk about C. elegans and how it's important and, why, we might want to study it. I mean, maybe a couple sentences, not a whole page of content, just enough so that we can establish, that then it gives you the advantage of kind of getting your feet wet with some of these model systems. I think that's a good way to approach that. And so if you need more references or if you have ideas for references we can add to this, let me know. And it's just, just a way to get your feet wet. We have other resources on some of our other, in other parts of our website, for example, where you can get more information on this. So if you go to the website, you'll find more information on some of these model systems. But this is just a very basic, overview so that people get their feet wet. And then finally, we have links to biological data sets. So we have a link to the DevoZoo, the openworm movement database, which is something that is run by a different group in openworm, but they do C. elegans movement. And so we've actually used that in years past for some projects, because it's actually a very nice, it's a nice data set, it's annotated, and it's adult C. elegans moving around in different ways. So they've taken like images of different movements that C. elegans makes. C. elegans makes a series of stereotypical movements, and those movements then are classified. You can classify them using machine learning. You can understand more about C. elegans movement patterns. So those are things that you might draw from, especially if you're doing like machine learning. It could be a benchmark, or it could be something, you might do something interesting with with some of the movement data. I think Mayukh did some work with this, and a couple other people have, been interested in it in years past. So that's something to keep in mind. And then I have a description of some of the types of biological analysis that we might do with some of the data. So once we have the data, what do we do? And this is something maybe it needs to be fleshed out a little bit more. So we do a lot of different types of analysis with the data. We're interested in temporal analysis. So we just recently published a paper on periodicity in embryogenesis. We're interested in network science. We're interested in trees, differentiation trees, lineage trees. We're interested in topological data analysis, something we haven't really done yet, but it's an interest. So there are a lot of different things we can do with the data. And I should probably put in, to improve this, I should probably put in some examples of papers that we published, which I will write as a note, because that's actually a pretty good idea. So, the idea is that we do, we're interested in doing a lot of this machine learning stuff and this analysis of images. But then, we have to make a connection on the other end, which is to do really interesting types of analysis with the data that we extract. And so that's how we build a paper, is basically that two-fold strategy. We're going to extract a bunch of things out of images, and then we're going to put them into these types of analysis, and then we're going to show the world what we can do with it. It's basically the idea. So, if you want to get involved in that, that's a pathway to a paper. It's a pathway to something very, potentially very interesting. So, so let's, if you have suggestions for this onboarding guide, please make them available. I'm trying to balance out having this thing be comprehensive without it being too unwieldy. So if we want to, I think there's the index here is a good start, but I don't know. We might end up splitting it up, if it gets too much longer. But that's okay because it's all hyperlinked. So that's the onboarding guide. Then I wanted to talk about DevoWorm or DevoLearn a little bit. So I said we're presenting on DevoLearn at the INCF conference, and I will talk to some of you about that. I know Mayukh will want to have some input, and I'll kind of put the slides together pretty soon. I did a short talk on this. Oh, thank you, Dick. There is a typo in there. I'll go back and check. Yeah, it's kind of hard to catch all the typos, but thanks. So I did a flash talk to another group on DevoLearn like about 2 months ago, and that went over pretty well, but that's a shorter version of this. I think we need to have like probably 3 or 4 times that length for this event. So I'm gonna add some slides in, and I'll pass it around to Mayukh and maybe some other people, and we can put together a really nice presentation. And I think this will be a nice showcase to the INCF people to show them that we're really building upon what they're sponsoring for us. So this is the DevoLearn repository. I don't know if we have a list of pull requests. We have one pull request that's open, but we've had a number of pull requests this week. So we've gotten a lot of contributions from people, potential GSOC aspirants here, Verdict, Mainak. And I think I mentioned this in the email last week that we've had some contributions. We've also had contributions to the Digital Basilaria repository. And that was, we had Shruti, we had Yash, we had a couple other people contribute. So this is a nice way to, again, get your work out into the world. And I don't know if Shruti's interested in pushing her cellular automata. So the DevoLearn platform isn't just the DevoLearn software. We have other types of things going on. And so we have the DevoWorm organisms. These are the model organisms, the data science demos, the general biological model. And I'm not sure if we have one set up for other types of models. I guess maybe I want to check before I suggest this, but I was wondering if Shruti was interested in pushing some of her cellular automata stuff to a repo that I will make here, and we can put it into this as a repo for other types of models that are outside of sort of the standard machine learning type of model. This general biological models is something Krishna proposed, and I made that separate. So that's there, but I don't want to put it there. I want to put it somewhere else. So, Shruti, if you want to get in touch about that, we can. I can incorporate it somehow into this place. Somewhere in here, I'll figure out where to put it. But
yeah, sure, we can do that. We can make a different repo for it or something. Because I'll be having more stuff too on Cellular Automata itself, so it would be good if we have a repo.
Yeah. So I think that's. Then I wanted to go over maybe some of our issues that we have on our board. Let's see. Oops. Projects, major tasks. So again, we have, this is, I know Jesse's been helping to organize this, but there's still a lot of stuff in here that's, we need to update it a little bit more, because things are always moving, so we don't know. We don't know where things stand. We have a lot of different, if you want to go to this project board, you can see some of the issues that we have. We have a lot of things. It's almost overflowing with things all the time. And so if you want to get a good sense of what kinds of opportunities we have, especially outside of GSOC, I would go to this board. Let me put a link to it here. So this is a GitHub project board. And, we have our different issues here. And these are GitHub issues. These aren't necessarily related to code-related tasks. They're just like issues that we have open. So we have the evolution conference presentations, for example. This is requires submission of an abstract by one of us, or the revision of an abstract. And so once that's done, then we move that to say like finished. We have other issues like update DevoWorm ML lectures. That's not something necessarily that will get done in one shot. It'll be like a gradual thing. So that will stay in action items until it's finished. And then, we have our DevoWorm bibliography. This is the EndNote bibliography. We also have this idea of a annotated bibliography, which is where you have a set of references with some notes underneath it, and you collect it, which is also something that I wouldn't suggest being a very general resource, but if you're interested in doing a deep dive on a topic, that would be a good thing to produce. You can do annotated bibliographies on different techniques or on different topics in developmental biology, or just a very specialized topic. We have something called complexity measures, which is very general. It's basically, we're trying to develop measures for looking at complexity in biological systems. So we have the Mathematics of DevoWorm poster that I showed 2 weeks ago maybe, where we have all these different high-level ideas, and then we try to reduce it to some of the mathematics that we're talking about in the meetings. And Jesse was interested in that. I know he's on this complexity measures issue, so this is still something in progress. I haven't had any chance to work on it since I showed it to you, so then we have things like getting data from different places, like Susan's ball microscope, and she's obviously working on that, as she said before in the meeting. When we have other things that are, so we have like this category of things that are off the radar too. So if there's something, we've talked about a lot of things in the meeting, and some things kind of fall off the radar, some things get revived, some things get sort of in the to-do list but never move out into the other categories. So if you're interested in grabbing an issue, or think about think something is interesting and want to follow up on it, please let me know. You can just put the issue number, in a Slack message or email and say, I'm interested in this issue. Where are we with it? And I can kind of tell you. So we have all sorts of even tutorials for YouTube. If you're interested in doing a tutorial on some topic and, recording a short video of yourself presenting on it, we could put that up on our YouTube channel. That could work. So there are a lot of ways to contribute here. And we don't have, it's purposefully busy because there's a lot of stuff going on. But, so that's our project board. And I know sometimes I go through the issues one by one, but sometimes that's not particularly effective because it's kind of, oftentimes we just have things that, we're following up on that. Okay, so then we have something in the chat. Bradley, Shruti and Krishna are working with me on the fish ladder toy model for a thermodynamically at equilibrium origin of life in a lipid world. Could be added. Okay, so this is the fish ladder toy model, thermodynamically at equilibrium origin of life in a lipid world. Do you have any smaller shells like you showed earlier? That's Susan to Dick. Let me add that to the list. Let's see. Let's see, how would I summarize it? The fish ladder toy model of life in a lipid world. I think we mentioned this before. Or we talked about this in an earlier meeting, so so this is a project issue. So this is something that I don't know if who I can assign this to, who's in the list of contributors. So if you're like a member of this, it brings you up. I don't know if I can issue it to people here. Oh, assignees, yeah. I don't have any of the people on the list, but that's okay. If you want to join this board, you can become a contributor. You just send me a request in your email address, and I can set it up. You have to invite people to the repository, and then I can add you as a collaborator. So that's actually pretty simple. And then I can assign you to different tasks, and you'll get an email on it. Okay, so project to be started, pattern resumption after self-repair in seashells. I can add that too. I know we did this, I think we talked about this a couple years ago, doing this type of project, but we could never get it off the ground. We had talked about putting it on a rig. I know I talked about this in one of the meetings recently. But this is actually going to be like something we'll get a lot of data on at this point. So I made 2 projects here based on the issues in the chat. Jesse says origin of life projects are interesting. Do you need our email IDs? All you need to do to become a collaborator on the project board is to send me a note with an email. I can invite you via email, and then you'll receive an email invite, and you become a member, I guess, of that repository. So you can be assigned to tasks, and I think you can make your own pushes to the repository. So that'll be a good way to get people involved in some things here. So I wanted to move on here to our papers for the week. So we finally have some time here to do papers, and I'm going to go probably a little bit over the top of the hour. So if you have to leave, that's okay. This will be recorded so you can get access to the full paper review once we, if you miss out. So I found this infographic this week. It's called Building a Cell in 3D. And I thought it was really interesting because we do a lot of analysis of images, and we're interested in 3D modeling, but these people have built a whole cell in 3D. And so they're showing this pipeline of how they've built this 3D model of the cell. And as you can see, it's really complex. So you can build a model of a cell as just a sphere, or a sphere with like maybe a couple of parts, like a nucleus, or maybe some other types of things in it. So if you're doing modeling, you know that like it's a matter of what level of abstraction you want to pick to model. So you can model a black box, just a shape that looks like a cell, and you might be interested in how it moves in its environment. And so that's one way to do it. But also in doing so, you might need to model things like different types of things in the membrane. So you might include those things. You might include the nucleus for whatever reason. But what they're doing is they're trying to build what they call a realistic model of the cell, or a high fidelity model of the cell. And so in this case, what they're looking at is they're constructing the whole cell 3D model. So they have this pipeline where they do this protein structures reconstruction. They have a recipe for generating this. Every protein is assigned a structural representation. They gather and validate structural data for each protein. Structures are retrieved from structural databases, are generated via homology modeling. The final recipe includes 482 protein monomers and 201 protein complexes. And they're actually, they're not working on a human cell. They're working on this Mycoplasma genitalium's bacteria. So this is a very simple cell as compared to something like a human cell or any sort of eukaryotic cell. So it's it's not even with like something that's a fairly simple cell, at least as we think of it, it's actually quite complex. So you have this protein structures reconstruction, modeling the nucleoid architecture. So now they're looking at modeling nucleic acids. They use this lattice-based method. They're using, they're modeling the genome of the bacteria, which is much simpler than ours, but still requires a lot of work to model it. And then they simulate the 3D organization of the cell. So now with the other two parts, then they put it into this, they assemble it and visualize it using something called CellPack. And so the CellPack, they use this software to stitch all this together into this 3D model. Recipe components are distributed around the nucleoid and relaxed within a defined cell volume using NVIDIA Flex, which is a specific type of NVIDIA technology. Ingredients are approximated to beads and subjected to relaxation or to resolve overlaps and clashes. So they just kind of like, they collect the data, then they put it, they take that, they put it into this model. And then they kind of try to infer the gaps in the model. So, when you take measurements in the real world, sometimes they're inconsistent, sometimes they're incomplete. And so then when you get like something that approximates what you're looking for, then you can make predictions to try to fill in the gaps, and you end up with this type of model. So this is actually, so then they say, well, you can explore the cellular mesoscale. So they're looking at using different color palettes to look at different features in the cell. So when you look at this thing here, it looks like you can't make any sense of it. And indeed you can't if you're just looking at it. But one of the things they do here is they use color coding to pull out different features. So this is, for example, looking at copy number variation. This is looking at structural confidence. And then this is looking at the source of structural data. And they have a legend underneath here to tell us where, what these mean. So, that's the way you end up, you end up building this huge model, and then you have to figure out a strategy of how to navigate all those data. Another way to navigate it would be, of course, to let people explore it. And if we're talking about the GSOC project on axolotl embryos, one of the goals there would be to build a 3D model or a flat map of a 3D surface and explore it. And not only explore it, but use these kind of strategies to sort sort out the data, and so that people, when they're looking at it, they can interpret things and they can zoom in on different things that they're interested in. So and then they just show the examples of how you can isolate different features in this model. So these are also color coded. So if you're interested in very specific genes or other types of things in the model, you can find them. So this is a poster by, here are the authors here. This was a poster, I can't remember where I got this from. It was off the web, but it's sponsored by Scripps Research. And I will put the link in the chat. And we have, Dick also wants me to add to the board analysis of shape of polygonal archaea and shape droplets. Okay. I'll add that after the meeting. So Akshay wants to be added to the GitHub repo. Okay, and so ABD's leaving. Thank you, ABD, for attending. So that's the building is cell in 3D. Then we have some other interesting papers here. I think this is something that Dick sent me, but I don't remember if this is the paper, but it might have been another paper on organoids. This is called Long-Term Maturation of Human Cortical Organoids Matches Key Early Postnatal Transitions. And so this is a paper that uses these things called organoids, and we've talked about them a couple times in meetings past, where they're building these they have a bunch of cells that are developmental cells, stem cells, that get differentiated in a culture. And they can differentiate them into things that approximate organs like the brain or like other tissues in the body. And so what they're talking about here is they're talking about cortical organoids matching key early postnatal transitions. So the abstract is, human stem cell-derived models provide the promise of accelerating our understanding of brain disorders. So they're modeling the cortex in the brain in mammals. And this is the area of the brain that we often associate with cognition and other types of behaviors in invertebrates. But not knowing whether they possess the ability to mature beyond mid to late fetal stages potentially limits their utility. So they can build these stem cell derived models in culture already, but it's hard to really make the link from that to like tissues that you might want to study. So we leveraged a direct differentiation protocol to comprehensively assess maturation in vitro. Based on genome-wide study analysis, it means they did it in a culture dish instead of like in an organism. Based on genome-wide analysis of the epigenetic clock and transcriptomics, which is how genes are expressed and like epigenetic effects of that, as well as RNA editing, we observed that the three-dimensional human cortical organoids reach postnatal stages between 250 and 300 days, a timeline paralleling in vivo development. We demonstrate the presence of several known developmental milestones, including switches in the histone deacylase complex and the NMDA receptor subunits, which we confirm at the protein and physiological levels. These results suggest that important components of an intrinsic in vivo developmental program persist in vitro, meaning that there's this program that is set in an embryo that you can replicate in an in vitro model if you have it set up right. We further map neurodevelopmental and neurodegenerative disease risk genes onto in vitro gene expression trajectories and provide a resource and web tool. And it's this GECO, or Gene Expression in Cortical Organoids site to guide disease modeling. So this is pretty heavy-duty molecular biology and disease science or disease biology. But the lesson here is that you can build these three-dimensional cellular ensembles that are, self-organizing and that form these sort of in vitro structures that resemble tissue development. So they're not replicating development as it might be replicated in the body, in the embryo, I should say. But it's but it's something that we can learn lessons from. So, yeah. So what they do is they embed these cells into a matrix, and they have the matrix has nutrients and things like that, and they're able to grow it not just with a large number of cells, but with shape. They're able to assign a shape, and they're able to assign some geometry, and this is how they're able to build these organoids. So they're actually the first group really to sort of give a systematic unbiased functional analysis to demonstrate the maturation of these different, the structure as it goes through these different stages, which are parallel to what you see in an embryo.
Bradley?
Yeah.
A question. Was there any indication that they're doing any time lapse or anybody has organoids?
Let's see. I don't know what they have in this paper in terms of the figures. Let's see what they have in the figures. So it looks like they're doing, I don't think they're doing time lapse. I think they're just taking images at different days and they're looking at sort of the markers within it. So, yeah, there's no real time lapse. It's just kind of like...
Yeah, it would be interesting to correlate this with time lapse to see if there's any differentiation waves that occur in organoids and if they coincide with these differentiations that they're observing.
Yeah, it would be interesting. I mean, this is, I don't know what kind of, this is human stem cells, so it's, or human derived stem cells, so it's, a long time period, on the order of 600 days, they have this out to 600 days. I don't know.
It's okay, you just get a longer time lapse interval. Right. Or you do it so that if anything changes suddenly, you start the camera.
Right. So, I mean, yeah, and I don't know how these, the organoids are grown in these controlled environments. I don't really know what the details are with respect to how they, change out the nutrients in the medium or the matrix, how that's done, how the imaging can be done, if it's something that you can actually do time lapse on, or if it's something that you can I know that they're doing a lot of electrophysiology in these organoids as well, and that's something that you can do not in a time lapse, but in a time series. You can get a time series out of it. So this is an example of some of the gene expression in some of these structures, and these are the images of the structures. So you see that you have these structures of the geometry, so they kind of look like they might be embryos where the cells are migrating around and they have a shape and they have a geometry, and so that's just and these are just different genetic markers that they're using to look at the expression of different genes. And so when you see these fluorescent markers and they become intense in some of these images, it's just that they're looking at specific things being expressed in the cells. And so for those of you who don't have a lot of background in biology, that's sort of they'll do this for different markers, and so sometimes you have to do massive numbers of experiments to get some of these maps that they have, where they have 300 or 400 genes that you can select in a model that they have in an interface. A lot of that is these serial experiments that they have to perform to get this information. So it's there is still this bottleneck of testing things one at a time, and we tend to think about things in large scale data sets, but the reality is it's getting the data is a lot harder than you'd like to have it. And so they just kind of describe these different modules, which are different parts of the organoid, and they have so they look at the differentiation day, and they're looking at these different markers, which are these different colors. So these different color functions represent the distribution of these different things. So the black line are immune response genes. The white cyan is extracellular matrix. So these are just different genes that code for different things in the embryo. And then these functions are the distribution of these. So these dots are different colors, and then these lines are the sort of the average of these different dots over time. So you're sampling at different times of differentiation. And you can see that these, like in different parts, like the glial modules, the neuronal modules, they're just comparing all of those in that category. So they're using something called Go terms, which are just something that is generated for a certain gene by external groups. They look at like how the gene is expressed and they give it a term. And then that's supposed to tell you what it does. And it's an imperfect method, but they can basically then look at gene expression, sort it by these categories, and then give you these sort of trends over time. So, dark green, there's not a lot of variation here. There's this purple that goes up early. That's the axon guidance, neuronal migration, and GTPase activity. So that happens rather early. And then you have things like the pink. I don't know if the pink is in here. Salmon is glutamatergic synaptic transmission. So this is the formation of synapses. This is something that happens maybe at day 300 and kind of declines slightly by day 600. So, these are things that are happening in, say, like the human embryo in development. This is where the nervous system is put together, and you can see that there's a spike at about 100 to 300 days where there's a lot of activity going on, and it trails off slightly towards the end of their time series. So, it sort of mimics human development, but there are, of course, going to be differences in these two. And that's, the idea is to get a good sense of what's going on in development, so we can maybe study diseases, maybe like a disease state versus a normal state. And by disease state, I mean if you have like a gene for some disease versus not a gene for some disease, we can look at the differences. So there are a lot more images in this paper if you want to look at that and get a sense of what's going on there. Another paper I'll go into here is this paper, Wiring Up Vision. And this is an interesting paper because they talk about, let me read the abstract here. It's an interesting paper because it's actually written by some cognitive scientists and some like artificial intelligence people. So they're minimizing supervised synaptic updates needed to produce a primate ventral stream. And so this is actually something to do with deep neural networks. So after training on large data sets, certain deep neural networks are surprisingly good models of the neuronal mechanisms of adult primate visual object recognition. So this is in humans and in non-human primates. Our visual systems, we want to make the parallel between that visual system that can recognize objects and a deep learning network that can recognize objects. Nevertheless, these models are poor models of the development of the visual system because they posit millions of sequential, precisely coordinated synaptic updates, each based on a labeled image. While ongoing research is pursuing the use of unsupervised proxies for labels, here we explore a complementary strategy of reducing the required number of supervised synaptic updates to produce an adult-like ventral visual system. So what they're saying is that in machine learning we use labels and we kind of label objects, and then that's how we sort of work around. Like we see a cup, and a machine learning algorithm sees a cup, but it needs a category to latch onto to say it's a cup or not. Well, what humans do, however, is different. We don't really necessarily need the categories. The system doesn't break if we don't have the categories. We can actually learn the categories with some language, but the language is emergent. And so our visual systems can recognize a cup versus maybe like a table without labels. We can do that in infancy, and then we eventually learn the labels and attach them, and there's meaning involved. So it's a very different system. So they're exploring this strategy, and so they're looking at the adult ventral visual system, which in the brain is a series of visual centers, the inferotemporal cortex, and then of course we can also learn from behavior what the visual system is doing. Such models might require less precise machinery and energy expenditure to coordinate these updates. And so this kind of approach would move us closer to sort of this neuroscience hypothesis of visual system wiring and maybe improve our ability to recognize visual objects in the visual sense. So they relative to the current leading model of the adult ventral stream, which we've characterized this, so it's not like something they're starting off from scratch on. We here demonstrate that the total number of supervised weight updates can be substantially reduced using these three complementary strategies. So first, we find that only 2% of supervised updates, epochs, and images are needed to achieve about 80% of the match to adult ventral stream. Second, by improving the random distribution of synaptic connectivity, we find that 54% of the brain match can already be achieved at birth. So that means you don't need training. You can have an unsupervised 54% of like performance that you see in like an adult system is achieved at birth. And some of that has to do with what's going on in the embryo in terms of organizing the visual system, but also because we can operate without labels. Third, we find that by training only 5% of the model synapses, we can still achieve nearly 80% of the match to the ventral stream. So there's a minimal amount of training here as well. The training comes in, like in the human visual system, there's a lot of connections are produced at birth, and then those connections are pruned through these experience-dependent situations. So when you're interacting with the world as an infant, you start to see things and you start to manipulate objects, and in your visual system, the number of synapses become pruned down to something that organizes it around the world that you're experiencing. So you don't need to train the model as much using this type of an approach. So when these three strategies are applied in combination, we find that these new models achieve about 80% of a fully trained model's match to the brain, while using 2 orders of magnitude fewer supervised synaptic updates. These results reflect first steps in modeling not just primate adult visual processing due to inference, but also how the ventral visual stream might be wired up by evolution, or a model's birth state, and by developmental learning, a model's updates based on visual experience. So in my other group, we're talking a lot about development and artificial intelligence. And so, but I thought this paper would be interesting to this group as well, because we're interested in a lot of issues related to artificial neural networks. So we do this, so they're actually looking at, like, the connections between neurons here. So in our artificial neural networks, these are the weights that are updated, and in biological systems, these would be analogous to synapses. And so what they're able to do here is to look at they're able to model synapses using a sort of a computational neuroscience model. And they're able to look at those that connectivity with respect to some stimuli and make these sort of estimates that they're making in the abstract. So they talk about sort of how at birth synaptic wiring is sort of optimized to start to get you started in the real world. So you see this in squirrels, you see this in humans, you see this in macaques. This happens across different species that have a primate, or a mammalian visual system. And so Anthony Zador wrote a paper recently, last couple years, on innateness in sort of artificial intelligence or in deep learning systems. And he says that, maybe we need to have an innate component to these models in order for them to perform better. And so they kind of go into that. But can we build a model that starts up kind of like a biological model where you have this innate ability to sort of pick things out of the environment, and then, if you train it on things, it doesn't necessarily need a lot of training with labels. It just needs to interact with its environment, and it can do almost as good as the machine learning models that we have now. So they talk about pruning, which is where the synapses are selected within development to become more focused and specialized on stimuli. And this is kind of the approach that they're using. And so they kind of go through the literature on this. If you're interested in this topic, I would read this paper and look at some of what they're doing here, their contributions to the field. So this contributions from the field, they kind of go through this method that they're using and they have their code on GitHub. So if you want to try it out, you can try it out there. So they have so these approaches that they're using. They build models of the fraction of supervised updates that retain high similarity to the primate visual system. They call that brain predictivity. They just want to be able to match it up with the biological model. We improve the at-birth synaptic connectivity to achieve reasonable brain predictivity with no training at all. So they use this at-birth connectivity, which is just kind of a dense random connectivity. We propose a thin critical training technique, which reduces the number of trained synapses while maintaining a high brain predictivity. So they're using this critical period as a way to sort of prune this network. They're using I don't know if it's a critical period in the conventional sense, but they're using this kind of approach. And then we combine these three techniques to build models of 2 orders of magnitude, fewer supervised synaptic updates, but with high brain predictivity relative to a fully trained model. And so they're using this. I think their idea here is to build something that has reasonable sort of performance statistics, but also matches what's going on in the brain. So we wrote this paper on comparing artificial neural networks and biological neural networks, and I think this would be a good paper to add to that paper when it's revised, to review kind of what's going on here as sort of the state of the art. I know we just kind of wrote that paper it just kind of like carried itself on to completion, but I think we can also add, there's always stuff coming out on this topic every day, just about, it seems. And so, but I don't think there's been a review, which is kind of what our paper is. So they have some really interesting stuff in here that they're doing. And so I would definitely, especially Krishna and Jesse, to take a look at this paper and look at it and just kind of critically evaluate it. So we have some things in the chat. Susan, do you want to say something?
Sure, I'm just interested in the references on the paper. I gather I can get that later, but if it was in the chat, that would be helpful too. I don't know if you can do that, but
the references for what?
For the picture presenting.
Oh,
I just look for the date on the papers. Just the reference. It's okay.
well, I put the drive folder in the chat. So if you wanna, I think I have the permissions worked out. So if you want to go there and look, I can send you the paper too. The organoids paper, correct?
Yeah.
Okay.
The organ paper is the one that I'm most interested in. The other one's interesting as well.
Yeah. Okay. I'll take care of that. So let's see. We'll go up to the top of the chat here. So we have. Oh yeah, major tests for 2020 needs to be updated to 2021. But this is Jesse's Skype and email. Data on the paper. The data on the organoids paper itself is, I think it's brand new. I don't know. 2021. Yeah, March of 2021. So it's pretty new. So yeah, this area of organoids, it's only kind of been emerging the last couple years, and so it's kind of the Wild West in terms of methodology. But they're kind of converging on some specific methodologies, and so there's a lot to learn in this area. You can build these organoids that have like this, geometry and this, in the case of brain organoids, you can look at the electrophysiological activity, and it can tell you some things about what's going on in development, but also what's going on sort of when you build an organ from scratch. What does that process look like? So
I was going to look at their viscoelasticity. That was my goal.
Okay. Well, I can just send you the paper after the meeting. So Tharun had to leave. Thank you for attending. Dick says projections not considered in paper being presented. So this is a paper that Dick wrote with a collaborator. Vision begins a direct reconstruction of the retinal image of the brain sees and stores pictures. So that's something that, and I'm sure that they have like they're just starting on like one. I don't know. I don't actually know what data sets they're using. I didn't look at the list, but this is an area that people are starting to pick up on, where they're looking at like biological networks, and they're saying, can you replicate some of those things for artificial neural networks? Because it's, artificial neural networks, they've built them in terms of size, they've built them quite large, but they still have a lot of errors in performance that biological neural networks don't seem to have.
Bradley, let me explain what I'm talking about.
Okay.
In computer tomography, you pass X-rays through an object and measure how many X-rays come out. And the X-rays go along lines, which are called rays. Okay, and so that's the standard approach to computer tomography. Now, vision actually does something similar. In vision, each cell in the visual cortex has what's called a receptive field, and these receptive fields tend to be long and narrow. And so this paper is about how to reconstruct images from a set of receptive fields. Okay? Each receptive field is like a line across the image with a certain width.
Yeah.
So in other words, what you're doing is basically you have these elongated shapes across the visual field called receptive fields. And the idea is that the brain has to reconstruct the image from all of these linear samples of the image hitting the eye.
Yep.
Now the thing is, that's obviously also probably also occurring, there's some evidence for this, occurring at birth. And so the learning of the visual system may be based on these projections rather than on whole images being input.
Yeah. Yeah, that sounds pretty good.
Yeah.
Yeah. Yeah.
So, we didn't look at the development in that paper, but it may be worthwhile looking at it from that point of view.
Yeah. Yeah, I'll have to take a look at that. Thank you for posting that. Then we have Jesse said Zador and innateness. Genomic bottleneck at NeurIPS. There was a thing at the NeurIPS conference on this topic, but yeah, it's like I said, it's moving pretty fast. So we might actually look at this more, read Dick's paper and look at it more in future meetings. So and then Krishna says nice presentation. So thank you for your comments in the chat. So I think we'll stop for today on the papers. Did we have any other things we wanted to bring up before we go? Okay. Well, if there's nothing else, thanks for attending this week. I don't know if we have, I know some of you are meeting after this. Thanks, Ujjwal. And we will be, in Slack or, via email. And, let's stay in touch over the course of the week. So we have a lot of things going on. So thanks for attending. See you guys next week, everyone. Take care.
Bye.
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 Yeah.
Bye.
I'll send you the proposal.
Okay,
by
yeah.
tomorrow.
All right.
Yeah, have a
Sounds
good
good.
day.
All right, you too. Bye.
Yeah. Bye. Bye, Jesse. Bye, Ujjwal. Bye, Bradley.